I'm Rory. I'm also 19. Actually, Amos and I were like JC classmates. Yeah. So, yeah, I'll be another of your like another presenter for today. And if you're going for like the econ taster course later, both of us will be there.
Yeah.
Yeah. Yeah.
Yeah.
No, seriously, you all have any questions for us? Like anything you want to know about? Maybe you didn't go for like your H2 bio talk yesterday because it was really early in the morning. And you want to ask us anything about H2 bio? Just just unmute. I think yeah, you all can unmute, right? Yes, you can.
Hi, could I ask what's the difference between H2 and H3 bio?
That's a
Oh,
very, very good question. Roy, you
okay.
answer that. Thank you, Roy.
Okay. Actually, I haven't. Okay, wait. I'm like you can do H2 and H3 bio. There isn't like a difference thingy because H3 bio, if like I don't know if you went for the other talks, right? But H3 is only done like in J2. So if you have the grades that qualify you for like H3, then you can go for H3 bio. Um, I've not in HE bio myself. I wasn't in HE bio myself, but then I have friends who are in HE bio, and then there was a lot of essay writing, a lot of stuff related to um like basically how like uh wait like how animals or whatever stuff came about, you know. Wait, Amy, do you have anything to add on?
Yeah, so for
Yeah.
example, like um, bacteria and bacteria and what's this called? Viruses.
Mm,
And you learn about their origins, you learn about the
mm,
chloroplasts, the origin of chloroplasts, or the origin of like mitochondria, if I'm not
mm,
wrong.
yeah.
Yeah.
Whereas like for H2 bio we focus on what what function does it serve right now and like the structure of it right now. Yeah. Yeah. So like I guess that's like a slight difference between H2 and H3 bio. H3 bio there's a lot of writing also, a lot of essay writing, if I'm not wrong. Yeah. And a lot of like um self-study, self-researching yourself. Yeah. Yeah. Are you thinking wait, whoever asked that question, are you thinking of doing H3 bio?
No, I'm taking O levels and my friend wanted to ask, so I'm asking for him.
Oh, okay.
Can you see me now?
Yeah, I can see you now. Hi Amy.
Okay. Hello.
Hello. Okay, we wait for about 3 more minutes. Bah.
Okay.
Okay. Yeah, in the meantime, anyone got any other questions? You can just ask us anything about ourselves.
Um, I have a question.
Oh, hello.
Yes.
Hi, is it possible to take H2 or H3 bio if I'm taking combined bio now?
Yes,
Um,
it is. If you get an A, I think the prerequisite for combined science is an A for the subject in O levels. Yes, it is. You are what's this called? You are not really allowed now, but you can. As in, there's no allow, not allow. It's just you can or you cannot. But yeah, you can take what's this called? You can take H2 bio if you're doing combined science in secondary school. Yes. Then
But
H3.
will the
Okay,
gap
sorry.
but will the gap be very big?
Uh well, the gap is very big. Honestly, it is very gradual how they teach you. So there will be a gap. There definitely will, but will it be very big? Not really, because I feel like what they teach you in O levels and what they teach you in A levels is it's not very different, but it's different. So, for example, like you learn the circulatory system in O levels, right? Yeah, none of that exists in A levels anymore. It's all like, yeah, it all gets lost somewhere in between O and A levels. You know, somewhere between Sec 4 and J1, it just gets lost.
mm.
Stuff like that, lah. Like the heart, also, you don't learn that. Dialysis, not dialysis. The what's this called now? You know, the loop of Henle and
Or
the
like the renal system,
correct.
like
None of that.
anatomy, all this. No, we don't learn that at all.
Correct. None of that anymore. Yeah.
Yeah. If you all have any like question, oh yeah, cause Amias was in the O level stream, whereas I was in like the IP stream. So, if you have any question, you can like just ask lah regarding O level IP.
Yeah.
For like context, we both did BCME or for H2s in JC.
Yeah.
Yeah.
Even though some of us may have wanted to drop to H1
Oh,
for like some subject. I mean, I don't know.
math, huh?
What? Guys,
What?
study hard in JC, okay? Don't
Okay.
give up on any subjects.
Yeah,
Study hard.
just press on. Okay, I think we can start soon.
Okay, I think we'll. Okay, yeah, I think we can start. Okay, hi guys. We'll do an introduction again.
Okay.
Now that I have video. So hi guys, my name is Amias. I'm 19. I was in the same class as her, and we both took BCME, Biochemistry, Math, and Econs.
Mm.
And I'm 19 this year. I'm in the army now.
Mhm.
And then she'll introduce herself now.
Okay, I'm Royi. I'm 19 too. I'm currently in my first year of university. And yeah, I took BCME. Oh, we're both from RI. If yeah, we didn't mention it, right? Yeah. So okay.
You want to tell them what you're studying?
Oh, I'm reading medicine in university. So if y'all have any questions, can like ask me that at the end, pa. Yeah. But then without further ado, we can start on viruses, taster course.
Okay.
Yeah. Okay. So like to start, we wanted a taster course. We wanted to choose a topic that would give like a good representation of the difficulty of like H2 biology. Because if we delve into like carbohydrates or lipids, it's something that we have covered in like secondary school or like something that's quite easy to grasp. But then we delve into like something really difficult like evolution or infectious diseases, that will also like confuse you. So we thought viruses would be a good like middle point. And it can be quite confusing during this taster course later. Don't worry because it's supposed to be confusing. When both of us did viruses, when we studied it in like the first semester of year 5, it was just really difficult. We had to do a lot of self-reading and like revision ourselves. So if you don't get it right, like you don't understand it like the first time, it's completely fine lah. Yeah. Okay. So yeah, let's start. And then even next slide. So are viruses living things? I think you included. Wait, can you click 3 times? Yes. Okay. So are viruses living things? This is a question that actually very few of like it's just we've never really thought of it, right? Like we know that we are living things. We know like bacteria is living thing, but what about viruses? So to answer this question, we need to like first consider what makes a living thing. In the next slide. and and to answer that question, we look to cell theory to figure this out. So what is cell theory? There are 4 main characteristics that like determine what a living thing is based on cell theory. And the first is that cells are the basic unit of life, and all living things consist of cells. So whether you are unicellular like bacteria, or like whether you're multicellular like us, um, essentially the basic unit is like a cell, and therefore we're all built based on cells and we're all living things. Okay, next slide. Secondly, metabolism occurs within cells. So metabolism, wow, big word. Basically, it just means that like you use energy to carry out processes that keep the cell alive, keep you alive. Stuff like respiration or like excretion of metabolic waste or like acquisition of food, etc. All these are important to like keep the cell alive lah. Okay, yeah. Another characteristics of living thing is that cells can reproduce, regardless of whether they reproduce by sexual reproduction or asexual reproduction. Either way, the cell is capable of dividing into 1 to 2, 2 to 4, 4 to 8, and like yeah, if you learn like mitosis and meiosis, you have like a parent cell dividing to form daughter cells. So living things are capable of doing such reproduction. Yeah. Okay. Next characteristic. Okay, and our final one is that cells contain hereditary information. Like in our cells, we have like the nucleus, which stores like DNA and genetic information of the cell, which will pass down to like our offspring. Yeah. So, moving on to that, like after we've considered what makes a cell, what makes a living thing, we need to consider whether like viruses fit like these characteristics. So our first point was that cells are the building unit of life, and all living things consist of cells. However, viruses do not have cellular structures, and they lack cellular organelles, which means that they are acellular and they are not considered cells. So this definitely challenges cell theory. Okay, next slide. Our second characteristics is that metabolism occurs within cells, but viruses they do not undergo metabolism because they are unable to respire, and they're unable to like feed and like excrete waste and stuff, and therefore they do not carry out metabolism, and this challenges cell theory. our third characteristics, this is like where things get a bit tricky because we said that cells can reproduce, like one cell becomes two, two cells become four, etc. The thing is that viruses by itself, they lack the ability to reproduce independently. But then when they're like inside a host cell, i.e. when like a virus goes into our body and then attacks our cells, when they're inside our cells, they can use our cells like proteins, enzymes, etc. to direct their own cell replication and reproduce. So by itself they cannot do it. But when they have like a host cell, they have a medium, they are able to do it. So there's like a question mark because can they really reproduce? So it's like a bit iffy. And like our fourth characteristic is that cells contain hereditary information. Okay, it is indeed true that like viruses are able, like they possess their own genetic material and thus they're not able they're able to like propagate like their genetic information to their offspring. And in from this aspect, viruses like this supports cell theory. So we've seen how like we have four characteristics. Viruses do not hold like they don't like it just goes against cell theory for the first two. The third one is a bit iffy, and the fourth one is support cell theory. And yeah, so what is the conclusion? Okay. The conclusion that many scientists have like come to is that viruses are obligate parasites, which means that they depend on like their whole cell to oh my god wait they depend on their whole cell to like complete their life cycle. So by itself independently they're unable to complete their life cycle, but then in that whole cells they are able to. Yeah, I hope this was clear enough. Does anyone have any question? Can you just unmute and ask?
Does that mean that they don't actually answer like yes or no to whether or not a virus is a living thing, but they classify them as something else instead and just avoid the question?
Yes, indeed, yes. So yeah, actually, because it's just super iffy whether they are considered living things or not. So instead, they just give it a special name, which is like obligate parasites. So yeah.
So it's an interesting thing, right? So next time when people want to talk about like, oh, COVID-19, blah blah blah, then you can flex, hey, COVID-19 is a virus, but do you think viruses are alive? And they'll be like, yeah, of course, what they'll be alive. If they're not alive, then how can they jump around and infect other people? And you say, hey, actually, they're neither alive nor dead, right? They are actually obligate parasites. Kind of cool, actually, right? Because I bet most of you didn't know that coming into the lesson. But of course, this is not the end of the lesson, right? So the next part of the lesson, we're going to dive more into, or actually we dive into the content. What Roy has just covered is the introduction to the topic. It's
Yeah.
the pre-lesson lesson. Okay, so now we go into the actual topic. Okay, so in our syllabus, H2 bio syllabus, we'll be talking about different types of viruses, right? And what we learn in these different types of viruses are that there are two main types. So the first one we learn are animal viruses. The next one we learn are bacteriophages. So animal viruses, they include your HIV, human immunodeficiency virus, and your influenza, which is your common flu, right? Next, we also have bacteriophages. Bacteriophage sounds something like bacteria. So basically, these are viruses that attack bacteria, right? And these viruses include your lambda phage and your T4 bacteriophage. You don't have to like memorize this now or anything. Your school will go through it, okay? It's just that we just name some examples for y'all to know because these are the ones that will be in your syllabus, okay? And so what do we learn about these viruses specifically? We learn about stuff like their structure, how they replicate, how transmissible they are, and how they transmit. Okay, we don't really go into like other things. It's more of the structure. The structure is really important, and the mode of replication is also very important. These are the highlights of your H2 bio syllabus. Okay, so of the ones that we chose, oh, it doesn't transfer. Do you see my next slide? Rui, do you see my next slide?
Your life cycle, the life cycle of influenza slide.
Yeah. Do you see
Yeah,
it?
yeah, I see it.
Huh, I don't see it. Okay, never mind. Never mind. Okay, so, um, so, okay, sorry. The life cycle of influenza. So why we chose influenza was because it is one of the more simple ones of course, your bacteriophages, right? Your bacteriophages, the ones that attack the bacteria, they are the easiest to remember. But what's this called? The animal viruses are harder because they're more complex, right? To infect an animal and to infect a bacteria, you can kind of guess which one is more, complicated. So influenza is the moderate one, okay? So influenza, the life cycle of an influenza virus, you can look at the A, B, C, and E parts of it. A, B, C, D, E parts of it. Okay, so we'll dive into it first. Let's go. First, we have attachment. So what attachment refers to is how the virus from the outside attaches to your cell, right? So these include stuff like you breathe in the aerosol droplets, right? Like for COVID, we breathe in the aerosol droplets that contain the virus, right? So once the virus enters our airway, right, and then it goes inside our body, it will start attacking our cells. That's how we get infected, right? We don't just magically get infected; they attach to our cells. So how they attach to our cells is there's this thing on the virus. You can see the first bolded and underlined word. It's called hemagglutinin. Very complicated name, right? But yeah, stuff like this you have to remember. Okay, hemagglutinin in your syllabus. So hemagglutinin is this thing that's found on your virus cell surface. Okay, so it binds to your sialic acid receptor. Wow, so complicated, all these terms, right? Sialic acid receptor. So this is found on your own cells. Okay, it is found specifically in the epithelial cells lining your respiratory tract. Okay, this is why when you get a flu, you only get symptoms in your respiratory tract, right? Because they infect that part of your body, right? So your hemagglutinin attaches to your sialic acid receptor, and once it attaches, the next one is that it goes through this thing called endocytosis, another really chim term. But endocytosis is basically your cell surface. Let's imagine it as a flat line, okay? So my head is the cell, okay, and the virus. So it goes in, and then it creates this bend in the thing, right? In your own cell membrane. Then eventually you can see that the virus goes inside your cell by this process called endocytosis. Okay, so it enters the cell by endocytosis. Your cell membrane invaginates, and the virus is placed in an endocytic vesicle. You don't have to understand all these terms now. We're just trying to explain the content. Okay, next. What endocytosis is, right? You can try to picture it in this diagram at the top left. Right, basically this thing comes in, attaches to your cell membrane. Your cell membrane invaginates, creates a little hole, and then after the virus goes inside, it will encapsulate it in a bubble. And that bubble we term a vesicle. Okay? Then there are other types of ways that viruses can enter, or that other particles can enter your cells. And there are fusion, exocytosis, budding. Okay, these different types are ways that things can enter and leave your cells. Next, we have this thing called the We have this thing called the endocytic vesicle, which was what we were talking about. Right? It fuses with this thing called a lysosome, another very chim term. Basically, it fuses with this thing that contains acid. Okay, so acid we know is corrosive. It's like it melts things, right? So because it fuses with this thing that contains acid. Now your cell membrane, or not your cell membrane, your virus membrane starts breaking down, right? Because of this lower pH, this acidic environment. And then whatever is inside your influenza virus comes out naturally, because whatever is holding it in is now gone. Now everything that's inside comes out. Okay, so the lower pH causes your viral envelope to fuse with the vesicle membrane, releasing the nucleocapsid into the cytosol, basically releasing the everything inside outside. Okay, next. Now this thing we see here is RNA, and we have touched it in O level before, right? We learned mRNA, right? So basically in viruses, their genetic material is not like ours, right? Ours in humans, we have double-stranded DNA, right? You learn our helical structure of our DNA in transcription and in DNA replication in our O levels, right? But in viruses, they can have DNA or RNA, and they can have double-stranded or they can have single-stranded. So they can have double-stranded RNA or single-stranded DNA as their genetic material. It's dependent on themselves, what they want to have as their genetic material. Okay? Of course, the story is more complicated, but let's just think of it that way first. Okay, so this special thing that influenza has is called the negative strand RNA. So what is this thing that this guy is talking about, negative strand RNA? We'll dive into that later. Just know that it is negative strand RNA. Okay, so your negative strand RNA comes out of your influenza virus, right? And then it now enters the nucleus. Okay, you don't have to care about all those other words, but it goes into the nucleus now. Okay, so once inside the nucleus, your negative strand RNA, because it's negative, right? Negative things we don't want in our lives. So we want to make things positive. Okay, so we have this thing, this enzyme called RNA-dependent RNA polymerase. So basically, just means that this enzyme changes the negative strand RNA into the positive strand RNA. Okay, so your negative strand RNA, we don't like negative things, we change it to positive. And only when it is in the positive strand can we make more negative strands. Okay, it sounds very, very complicated. But basically, we have negative strand RNA, that is what the cells are made up of. But in order to make more negative, we have to change it to a positive strand RNA, then change it back to a negative strand RNA. Okay, very, very complicated, but you'll get the hang of it once you dive in. Okay, because this is not one of your first few topics. Okay, it may seem hard, it may seem like, what are all these terms he's throwing around, right? Your school will teach you this entire topic in what, 2 weeks. Okay, we're trying to cover it in 1 hour. So of course, it's gonna sound like, what is this guy talking about? Why is bio so difficult? Okay, remember that this will take about 2 weeks in school. We are doing it in 1 hour and less. Okay, so bear with us if you don't understand things. Just to give you a picture of how the content looks like. Okay, next we have our complementary RNA strand. So now this is our negative RNA strand, right? It moves into your cytosol, which is basically your cytoplasm, and it serves as your mRNA. Okay, so next, because you have mRNA, right? And then we all know what mRNA does based on O-level syllabus. It undergoes translation. So then it undergoes translation, and then it becomes, or it synthesizes viral structural components. So this include like your in this slide, your capsid proteins and your glycoproteins. Don't have to know what these things are. Basically, they just make up the cell. Okay, they make up the virus, not the cell. They make up the virus. So the virus, you can imagine, right? It has its DNA in the middle. It's surrounded by a protective layer, and outside the protective layer, you have other things poking out of it. You all know the COVID-19, right? Have you all seen the pictures of it, right? Or the picture that CNA, Channel News Asia, likes to have on their YouTube videos, right? It's basically these things popping out of the side of the, what's it called now? Of your COVID-19 virus. Think of it like a golf ball. Can you all imagine how a golf ball looks like? Imagine you stick like toothpicks on the surface of the golf ball. Yeah, things like that. Okay, that's your glycoproteins. Okay, so your virus, right, is now using your host cell, your animal host cell, to create more of itself. So after it has synthesized its genetic material, right, the next thing it needs is the things that surround it so that it can become actual viruses, right? So you will synthesize these viral structural components, your protective layer, which is your capsid proteins, and your glycoproteins, which are the things on the surface of your virus that helps it recognize other viruses, helps it recognize animal cells. Okay, so these are some of the things that are synthesized from the translation of your mRNA, which is your negative strand RNA, which is your influenza virus's genetic material. Okay, if all these terms are flying over your head, okay. Next, so your complementary RNA strand can also be used for the synthesis of new viral genome. This one we covered just now, right? Your negative becomes positive, positive goes back to negative, right? Next.
Wait, I think here's like a good place to pause first. Does
Okay.
anyone have any questions about like the replication process or anything before we go on to like how assembly of the viruses? You just unmute yourself. Don't worry, you just ask. Okay then I guess like Amy can continue but
Okay. So the next thing is that so after all these things have been made, right, they have to be assembled to form back your virus. Imagine your virus as a human being, right? So you create its DNA, you create its like heart, its head, its arms, but all separately. Now to actually have a proper virus come out of, to assemble a human, right, you need to put all these things together so that it can uh, infect other cells, right? From the virus point of view, it has to replicate itself, assemble all these parts that it has synthesized, and then come out of the cell and infect other cells. Because that's its main job, to infect other cells, right? So now we assemble all these things that we have created. So we assemble the new viral progeny, right? So your blah 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 associates with blah blah blah. Okay, because all these terms you don't need to know. So blah 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 associates with blah 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 becomes blah blah blah. And then after all the things come back together, it exits the cell. Okay, exits the cell by budding. So just now, one of the processes we talked about with budding. So basically, you can see in this picture, right? So the virus, at step number 7, the virus already assembled itself. Next, it is encapsulated in this small semicircle of a bubble. And then you see at step 8 is where the bubble releases from the animal cell. Okay, so next, the whole cell evaginates, right? That means that semicircle pops up, right? And the newly formed viruses leave the cell by budding. So you can see in the picture there at step number 8 again, you see the scissors symbol. That scissors symbol is this thing we call neuraminidase. It is a enzyme, is an enzyme that helps the bubble leave the animal cell because the bubble is made out of the animal cell's cell membrane, right? So it is technically still connected to the cell membrane. But how do we make this bubble leave the animal cell? We have to cut it. Okay, but in actuality, it's not really cutting because cells don't have scissors attached to them, right? So they use an enzyme to free themselves of this animal cell, and they can leave the cell by this process we call budding. Okay, so yes, neuraminidase is the enzyme that helps to facilitate the release. Okay, can you see my next slide? Oops. Okay, so this is the last slide we have. So what we have gone through up to now is the life cycle, the reproductive cycle of a animal virus that we call the influenza virus, right? Your common flu. Other life cycles that will be covered under your topic of viruses in your H2 syllabus include your lytic cycle, your lysogenic cycle, and your HIV life cycle. Okay, so uh, honestly, for me, the easiest one is the first one, lytic cycle. Then there's the lysogenic cycle, and I feel HIV is the hardest. Yeah, it's either HIV or influenza that's the hardest for me. But other
HIV is a
than
bit
that,
tougher because there are like
yeah,
more steps in between. Yeah.
correct. Yeah. So yeah, so the first two, like lytic cycle and your lysogenic cycle, these two are the easiest. And yeah, it won't be hard for you to learn them, but it might be a little challenging when you move on to your animal viruses. But again, your school will take 2 weeks. It will go at a comfortable pace, and just pay attention to your lectures and ask questions after lectures if you are unclear about anything the lecturer has spoken about. Which in your case now is now. So if you all have any questions on what Roy and I have just presented, you all can unmute yourselves and ask. And yeah, if not, uh, yeah, we have nothing else planned for this taster course.
Wait, actually, I created like a Menti to like collect questions.
Okay.
Cause
Yeah.
cause you are so shy.
Sounds
Yeah.
good.
So,
Okay.
but the thing is, I can't share my screen. Okay, but like, So like yeah, my
Okay.
the code is 52825105. Yeah,
Mentee.
5282. Yeah. Okay, I'll just read out the question from here, but like, oh
Five
my
two
god,
eight two
5105.
5105.
Yeah. So like you ask
Okay.
question. Wait, no, wait. Amy, you can't view the questions from here though. Oh, like yeah. Okay, like I can see the questions.
Oops. Okay.
Yeah, but I can't share screen. Okay. Anyway, the first question we got was like, what do you think was the hardest bio topic? We want to answer that.
Oh. Okay. You go first. You go first.
Okay, the hardest bio topic for me, um, let me think. Mm, I think evolution was quite tough for me
Hmm.
because like it's just so broad. They try to cover a lot of things at once, and there was just a lot of things that they could test in like exams and like and like, I think cause like evolution in our school it was done in like two parts, part one, part two, so they just made the content a lot more, and yeah, I don't have much to add on for that. If you want to talk about yours.
Okay. Oh, actually, you know what I should do? I should pull up
What?
the freaking H2 bio syllabus, right? So everyone can see
Yeah, okay.
what topics there are even in the first place, right?
True. Okay.
Okay. So um oh, I just realized they don't actually tell you the things. They just give you the learning outcome. So that was a okay. Wait, no, we have the four core ideas. Okay, the four core ideas. So basically, right, um, in your first one, they teach you stuff like your carbohydrates, proteins, lipids. Those are very simple, right? And with your proteins, you will also learn enzymes. And what else is there? It's right here. Okay, you have cell structure and membranes also. So these are just very simple, very simple basic topics that you'll be taught at the start. Because remember, it's progressive learning, right? A levels. They don't teach you all the hard stuff at the start. They slowly bring you into the hard topics. So yeah, so the first few topics, those are very simple. I don't think you struggle much with it. You will struggle at the start maybe, but after a while you get the hang of it. Right? Then next core idea is your genetics and inheritance. This will include things like your mitosis, meiosis, DNA genomics, which is your DNA replication, translation, transcription. It will also include your things like your viruses, your bacteria, and your pro and eukaryotic genome. So what is pro and eukaryotic genome? This is what will sound unfamiliar to you all, right? So basically it's just the difference between your animal DNA or your multicellular DNA and your acellular DNA, something like that. It's like a rough explanation of how it's gonna go, right? So we talk about things like how the DNA differs in terms of structure, how it differs in terms of other more interesting ways, okay, more interesting characteristics, how it would differ. And also in that topic, you will learn about cancer, which is one of my favorite topics. So sorry, getting back to the question, because I always go astray. The hardest topic for me will be core idea number three, energy and equilibrium. So in energy and equilibrium, we cover things such as let me pull it up, your respiration, your photosynthesis.
Yeah.
Okay, and for me, the hardest topic in H2 syllabus is what's it called now? Photosynthesis.
Oh, yeah, because there were a lot of like terms and like processes and random words and random like enzymes and a lot. Oh yeah. Okay, photosynthesis and respiration were really tough because like there were a lot of chemical pathways. Then you had to
Oh,
actually memorize the enzyme. So like
hmm.
what was it? Oh my god, so many all like the intermittent.
GFG13 GPA OC
Oh yeah.
alpha.
Yeah. It's like
Yeah,
glucose, 1,3 bisphosphate, all this.
yeah.
I don't remember many
Okay,
of them.
but it will
Yeah.
make sense when you study it because all the terms you will
Mm,
learn it right in at almost the same time you learn organic chemistry. So when you learn your organic chemistry, you understand how the chemical compounds are named, right? And then when you understand how they're named, and then you see the names of the biochemical compounds that you are studying in respiration and in photosynthesis, it will make a lot of sense. So it won't be hard to remember. It will just take you a bit of time to internalize, right? Oh, also another one that is difficult for me, also under energy and equilibrium, will be cell signaling, right? Yeah, it will be
Oh,
cell signaling.
okay, yeah, because that
So
one has just
basic,
a lot of things to memorize
correct.
too. Yeah.
Cell signaling, for me, why I struggle with cell signaling and photosynthesis is because these things are more abstract. These things I don't really see in my day-to-day life. Okay, but not that I see other things lah, but it's just harder for me to understand photosynthesis and cell signaling. Cell signaling is basically they tell you how your individual cells react to chemical signals. For example, we all know insulin, right? We learned that in homeostasis in O levels, right? So we learn like how insulin, um, when it is attached to the cell, how the cell then undergoes so many processes to have a reaction. To have a reaction. Yeah, so that's what's hard for me. That was those two are the harder ones for me. But a lot of people also struggle with, you see part two, the extension topics, infectious diseases, right? A lot of people struggle with infectious diseases because it is a huge topic. I would say it is the biggest topic in bio. Maybe it will be tied with evolution, but infectious diseases is so big. Okay, but if you ever struggle with infectious diseases, right, you can check out our YouTube channel, because I did a video where I went through infectious diseases, right? It is a 3-week, maybe 4-week kind of topic. I think they took 3 or 4 weeks in school to have this, what's it called now, to go through this topic. But I did it in 1.5 hours, I think. But only watch it after you've learned the topic, because then it'll be easier for you to not learn after you have gone through the lesson. So you'll be able to internalize everything in that 1.5 plus hours. Yeah. And I go through questions also. They're like a typical question. So yeah, if you all ever need help with bio, you all can check out our videos at our YouTube channel, because we go through the more difficult topics. And yeah. Okay, any other questions?
Like I think there are like 16 questions. Some of them
Oh no.
Emias has already touched on, like what are extension topics like. So I'll just mark
Okay.
that as answered. There are a lot of questions about our future plans and medicine, which we will touch on like at the end lah. Okay. There's regarding like content related regarding today's data call. There's a question on what's the difference between budding and exocytosis.
Okay,
Emias,
that's a very, very good question.
you want to pull out that picture?
Yeah, sorry, I just need to
Just fine.
share my screen again.
It's actually both are involving like the thing exiting from the cell. So unlike endocytosis and like fusion, which involves like entering the cell, exocytosis and budding involve like leaving the cell. And if we look at like that picture from earlier,
Pulling up. Hold
wait,
on. Sorry.
give us a while.
Okay,
Yeah, okay,
here we are.
yeah. So you can see that like for exocytosis, the like assuming this is a virus, then there's an envelope. The envelope fuses with like the cell membrane, which is the blue part, and then releases the content outside. Whereas for budding, like the initial stuff doesn't have like an envelope, so it just like it, how to say it like takes up the cell membranes like part of the cell membrane. Do you like visualize it?
Okay, or you can think of it like so you have your virus. So your virus is a human being, and the cell membrane is the door of the house. So the virus that had a jacket on inside, because inside his house was very cold, when he leaves the house, he leaves the jacket at home and then exits the house naked. As in like with his shirt lah, as in without the jacket. But then when he's for budding, he is inside home. It's very hot at home, but it's very cold outside. So when he leaves the home, he has to wear the jacket. So he takes something from home with him as he leaves. Whereas for exocytosis, he doesn't have anything when he is. Sorry, he doesn't
Yeah.
have anything when he is
outside, when he's outside.
outside, but then when he comes back in, he wears something.
Like, yeah, when he's inside, he has something on. Then he ditches it, and like it fuses with like the door,
Correct.
and then
So
he
it
goes
stays
outside.
within the house.
Yeah.
Yeah.
Yes. Okay, so I'll just mark that as answered.
Okay, but if you still don't understand, please ask us to clarify.
Yeah,
again.
please continue
is-
asking
yeah.
questions.
It's just whether or not that part stays with him or whether that part leaves.
Hmm.
Okay.
Okay, there's another question on like how many topics are there in total. I think this one Amos has touched on already.
Hmm.
Uh,
But not
uh,
the exact number. I'm trying to pull up my own list if I
I think
remember.
this one like your school will go through with you at the start. But I think what's more important is the core, like the 4 different core like okay, yeah, here you go.
Yeah.
Here are
Okay,
the
so this was what I used to study this list. Yeah. Okay, so let's go through them. So water potential, it is the easiest topic ever. They don't even test you this. It's just more of a to understand future topics kind of topic. So water potential is very easy. Carbohydrates, you all touched base already in your O levels, but it goes, of course, delves deeper into it. Otherwise, it won't be called the A levels, right? So yeah, you learn about things like how your glucose monomers. So glucose is one building block. Glucose plus glucose gives you fructose, right? So it tells you how these reactions occur. Also dives into the food tests that we have learned in O levels as well. Lipids, same thing. Lipids is also okay. Lipids will be the second easiest topic in the entire syllabus. After water potential is lipids. Then carbohydrates. Proteins is not that simple. It's not as simple as you think it is because you learn carbohydrates, you learn lipids. You think, oh, how hard can proteins be? They're not that hard, but it is much harder than your carbohydrates and lipids. Okay. Then with proteins, you will obviously learn enzymes because these two are interlinked. Okay. Cell structure, you all have touched base in secondary school also. You learn about your organelles, your functions of organelles. Next is your cell membrane. So cell membrane, you touch on things like what we talked about just now. Your endocytosis, exocytosis, budding, and tell me of the last one, Roy.
Endocytosis?
Endocytosis, exocytosis, budding,
Fusion.
and fusion. Yes. Yeah. Okay. So this is actually how we study sometimes. Like, like when we study in school, whenever she has a problem, right, then she'll ask me. Then whenever I go, I'll ask her.
Oh,
Yeah. So
actually on a like okay, not like a related note, someone also asked, do you have a timeline of the topics that were taught in J1 and J2?
okay. So yeah,
yeah.
we can go through that also.
I
Okay.
can wait, or you go. Oh, okay, okay. So like if I remember correctly, we learned everything up till viruses. That was like from sem one of year 5. Yeah. Then bacteria to like bacteria to what Amy?
Should be photosynthesis.
Bacteria photosynthesis was like sem 2.
Yeah.
So by like so all these were covered in like year 1, which is quite a lot lah. Yeah.
Okay, but if you think about it, it is doable.
Yeah.
Because up to now, okay, let's see. So I have all the J1 topics in one screen now. Okay, from carbohydrates to photosynthesis. These are all the topics you're going to learn. But truthfully, what are things that will make up the most of your paper will be DNA genomics, both 1 and 2, your pro and eukaryotic, so that's the P and E that's written there, your respiration and your viruses bacteria. That will make up the majority of your paper. Stuff like your carbohydrates, lipids, proteins will make up a small little, maybe in your MCQ, will take up a big portion. But other than that, basically your DNA is a huge topic in your J1. So DNA, it can be found in your everything after DNA and genomics 1 all the way until pro and eukaryotic 3 is all have to do with genetic material. Yeah. And other stuff, of course, but mostly genetic material. Yeah. So, okay, going back to this. So mitosis, meiosis, cell division, we have learned this in what's this called now, O levels also. So not that difficult. DNA genomics 1 is basically just replication. DNA genomics 2, okay, so why there's a 1 and 2? I think only in our school do they separate it. Otherwise, in other schools, I think they just teach it as one bunch.
I think so too.
Yeah. So DNA genomics 2 include your transcription and translation. So you might be thinking, I've done all this in O levels already. Why do I still have to touch more on it in A levels? Because what you learn in O levels is just the simplified version.
Mm.
When you come to A levels, you learn about much more. Okay, so you know your ribosomes. You all remember your ribosomes? Yeah, your ribosomes. Correct. Yeah, you have two parts actually in A level syllabus. You have two parts. So your large ribosomal subunit and your small ribosomal subunit. So that's like one of the differences between O and A levels. But of course, there's much more lah. Okay, viruses we went through. Bacteria is also another topic.
Wait, Amy, for like the viruses part, right? Someone asked, is it 2 weeks for the entire cell theory? Like in terms of like, because you mentioned 2 weeks earlier. 2 weeks for entire
Yeah.
cell theory, including all the cycles, or like 2 weeks for each life cycle? For cell theory, I think they only spend like 10 minutes on it, in like, or like 5-10 minutes on it,
Yeah.
just an introduction. It's like the focus was on the cycles, and I think we spent 2 weeks for like the entire of viruses, if I'm not wrong.
Yeah. Not 2 weeks per cycle. 2
Yeah,
weeks
no, definitely
for
not.
all the
Yeah.
cycle.
I remember
Yeah.
like one lecture they went through like both, like one lecture they went through both HIV and influenza, if I'm not wrong.
Yeah, but it was a hard lecture to pass
Yeah,
through.
not
'Cause
easy.
all these terms the lecturer is throwing at you are just flying over your head, and you're like,
Yeah.
what is this lecturer talking about? Okay, but so a pro tip, right? Going into JC, a pro tip would be to read up on your lecture materials prior to actual lectures. Everyone tells you this, and you don't understand the like importance of it. I never understood. Okay, and one of the things I regretted was not doing that. Okay, because if I actually had read, right, I wouldn't have struggled as much as I did after lectures or like during lectures. Because if I had read up, like some of my friends did, the lecture materials will make more sense. They'll be like, oh, okay, this is why I read, but I didn't really understand because it was missing this explanation that the lecturer just clarified. Stuff like that. Okay, so bacteria is quite easy topic. Yeah, it's quite easy topic. Pro and eukaryotic 1 is very simple. It's just your structure and organization of genetic material. Pro and eukaryotic 2 is where it gets a little bit difficult. It talks about how do you say? It talks about a lot of details in your genetic material that you didn't know you didn't know. Okay, stuff like how they turn off and on some genes. Okay, how they turn on and off some genes within your body. That is what you learn in Pro and Eukaryotic 2. The most simplified version, the most simplified explanation I can think of. Pro and Eukaryotic 3 is basically cancer. Easiest topics, one of the easiest topics. Respiration, this is where it gets a little bit hard, but respiration is just a precursor to photosynthesis. Respiration is pretty simple. I would say it's pretty simple. There's also a video on it on our YouTube channel. Photosynthesis, this is where I struggled personally. But it is not that difficult, okay? It is really not that difficult if you just give it a shot. Mendel genetics, my favorite. Okay, one of my favorite, Mendel genetics 1 and 2. You learn about inheritance. You all remember your things like you all learned this in O levels, but how do I remember your blood groups? You know, you have your I and then you have your superscript A, I, B, I, O. You all learn in O levels, right? Same thing. Mendel genetics, same thing. It's just you get more in depth, right? You learn about things that are more in depth. You all learn in O levels. I remember. Molecular techniques. It's a very hard topic to start, but after you start, it gets very simple. Do you have an opinion of that, Marie?
Um, mm, I think my art teacher was quite good at like drawing everything out lah, which is why it was okay. So wait, I think we need to speed up. We have like
Okay.
10
Yes.
minutes.
Very fast. We're almost done.
A lot.
Stem
And
cells. These are the.
yeah,
Yeah. Sorry.
I think you can actually view the questions too. I just realized.
Oh.
Yeah.
Okay.
But
Yeah.
later.
Stem cells are basically these magical things that hopefully can save the entire world in the future. Okay. Cell signaling. These are things that they teach you how the cell responds to external signals. Right. Evolution. Evolution is basically
How it evolved from like like how did
right.
like all different species come about? How
Right.
did one species become different from another species? What is the concept of species even? Yeah.
Right. So infectious diseases, my favorite topic, teaches you about things like tuberculosis, how how your body reacts to any bacteria or viruses that comes and attacks you. Right, the entire process from when it infects you to when you generate your antibodies. Last topic that's not written here because it got removed from our
yeah,
from our syllabus is called climate change. That is one of the extension topics that's in the syllabus paper. Okay, next question, Roy.
Okay, I think you can go and pull it out. Like, do you have like the link to? Okay, but yeah, that's okay. There were quite a few questions about like why do you choose bio over physics, or like how do you make a choice between bio and physics? Let's read through this. For me, I chose physics because not physics. I chose bio because like I didn't like Sec 4 physics. There was a lot of like the electromagnetism and all those stuff, and I just didn't like it. And I just found bio so much more interesting, more applicable. I think physics is definitely very applicable to daily life, but like it's like something different when you're learning infectious diseases like alongside like the whole COVID pandemic, when you're knowing about the SARS-CoV-2 like virus. It's just very different lah. And like you can really see the real life applicability of like everything, which is like super cool. How about you, Amias? Why do you learn bio over physics?
Oh, I hated physics. Very simple. I just hated physics. Couldn't stand the topic. Couldn't stand anything that it stood for. I just hated it. That's why I chose bio. Yeah. It was a very easy. I feel like our conundrums. Oh, we didn't have a conundrum, but if you all have conundrum, I don't know. I can't really help with that because I just hated physics.
Yeah, yeah, same. I already decided on bio like way before. But if maybe your school has like introduction lectures to bio and physics, then you can sit in and like compare. Because I remember our school had. Yeah.
Yeah.
Yeah. Okay.
Did we do better in physics? Oh,
in
are these
bio,
people going for the econs? I mean the math.
I think so. Oh my god! Wait, we gotta like rap through this quick.
Okay, we speed run.
Speed run,
Did you guys do better in bio than physics in school? Yes, very much better.
I was about the same like. It's just more of interest. Okay. Uh, what are your wait? Okay, there's a lot of
What's
math
the difference?
questions. Do I answer the math question? There are like a lot, and it's like quite.
Okay, how about we create a freaking Zoom link and then we can move there.
Okay, also can.
Unless you only go for math. If you only go for math, you can stay here and go for math. But those who, okay, I don't know. Should we?
You just do it lah. Just do it.
Okay, can you create? I'm very technologically behind.
Oh shoot! Wait. Okay. Wait. Meanwhile, you answer other questions.
I'll answer other questions. What does Amias want to study after NS? Okay, Amias wants to study. What's it called? He doesn't know yet. Okay, that's a very good question. What's done in biopracticals in JC? What's done in biopracticals in JC? Oh, actually, very cool things. I really like biopracticals. Every time we have a lesson, oh, I'm so excited. I go into the lab and I'm like, okay, Mr. Prab was my teacher, our teacher. I was like, oh my god, Mr. Prab, what are we going to do today? You know, because wow, so cool. All the cool things, you know. Basically, you add this chemical into water, right? Then you have like photosynthesis happening, and then when the photosynthesis is happening, it's like so cool because the color changes, and it's like wow, you know. You also do your what's this called? Your food test. Food tests. I love food tests. Food tests are always so fun. Okay. Thank you for sharing. Just other things in practical. Honestly, other things in practical. I don't know. They're just all quite cool. There's microscopy. Yeah, basically, you just look at things under the microscope. Those are the like boring ones. You don't have much of those, so don't worry. What else are there? Cool lah. They're all quite cool. They're all like, oh, I never knew that I could experiment using these methods.